I mentioned earlier that RNA was made as a perfect copy of DNA, but then it had to be edited down. And that editing process is reflected in some more terminology that we're going to throw at you now called exons and introns. Exons are the expressed region of the DNA. In other words, that part of the DNA which is made into protein. Introns are the intervening region of the DNA. They're not made into proteins. They're the parts that are removed in the editing process. So in this diagram, the exons are shown in green, and the introns are shown in brown. And the DNA sequence, the gene, has both exons and introns in it. And we take several of these exon regions, exon 1, exon 2, exon 3, we cut them out and we splice them together to form the final messenger RNA, which is translated into a polypeptide or protein. In the same way that if you've ever done this, you edit your own work as you're writing a research paper or a term paper or a thesis. You take out words that don't belong. You take out sentences that don't belong. You edit those out to produce the final product. It's the same way that I'm making these videos, as I mentioned. So I'm taking out all my stumbles, all the things that I don't want you to hear, and I remove those, and that's part of the editing process. So those parts of the video which I've removed and you never get to see that were there at the beginning but have been taken out, we'll call introns of those videos. And the parts that you're actually seeing are the exons. So the exons are the parts of the gene that are actually made into protein. This is showing the same concept, but just a different artist. So it's the same information that you had on the previous slide, but just a different way of looking at it. And so either one is fine. You don't have to spend a lot of time looking at all of these. Just understand that whichever one it is that makes you see this process is fine. They all will take you to the same place. So in DNA, at the top of this diagram, I have exons represented by E and introns represented by I. I make an mRNA transcript, which is a complete and total copy of that gene. Exons, introns, trash, the whole thing. Then there are processing enzymes shown here by scissors within the nucleus of the cell that remove those introns. Those get recycled. I would say they're thrown away. They're not part of this message, but they are reused by the cell. Those are shown as the excised introns. Then we take the exons and we sew them together. We splice them together into the final transcript, which is taken out of the nucleus through the nuclear pore and taken to the cytoplasm where it can be turned into a protein. This process is called RNA editing. And the RNA protein particle that does this editing, the scissors that we saw in the previous diagram, is called a SNRP, SNRNP, which is short for small nuclear ribonucleoprotein. So the name tells us that it's small. We find it within the nucleus, which is where this editing is being done. It's a collaboration between RNA or ribonucleic acid and protein. So it's a small nuclear ribonucleoprotein, or a SNRP. And that's our RNA editor. And it takes out these loops of RNA that are called lariats. So the introns that are going to be removed become lariats. The lariat is the physical structure that underlies the concept of the intron. Those get snipped out and the exons are spliced together by these SNRPs. So the SNRPs are the scissors and paste responsible for this editing process. Once that editing process is done, then that functional messenger RNA can be taken out through a nuclear pore and taken to the cytoplasm. Well, I hear you saying, well, Dr. Hutchins, that's all fine and good, but has anybody ever gotten sick as a result of this process gone wrong? I mean, this is all this wonderful egghead theoretical stuff, but does it actually have any application in the real world? And you know, I wouldn't be teaching you this if the answer were yes. So now we look at the application in the real world of what happens when editing goes wrong. There's diseases which are fairly common in populations of Southeast Asia and in South Asia and in the Mediterranean regions and in sub-Saharan Africa. And they're called the thalassemias. These are blood disorders that result from the abnormal transcription and or translation of the genes for alpha and beta globin. Now you may notice that we're going to use the hemoglobin molecule over and over again. We started with it in Module 3, talking about the structure of proteins. Now that we're familiar with it and we've talked about the heme groups, which are the iron-containing oxygen-carrying groups within hemoglobin. So hemoglobin without the heme group, just the protein part, is called globin. And there's two different forms of globin, remember, forming this quaternary structure of the protein called alpha and beta globin polypeptides. These come together to form one complete hemoglobin molecule. Well, in the thalassemias, these globin polypeptides aren't made right. And if it's the alpha globin polypeptide that's not made right, we call it alpha thalassemia. And if it's the beta globin polypeptide that's not made right, we call it beta thalassemia. And beta thalassemia turns out to be caused by abnormal splicing of beta globin. I'm going to go into the details of beta thalassemia, not for you to know for the exam, but only if you're interested in. So this is another segment that you can skip over if it's not something that you're particularly interested in. But in beta thalassemia, there's a mutation in DNA. A T in DNA is changed to a G. At the same time, then, in the RNA transcript, a U is changed to a G. That puts in a splice site that doesn't normally belong there. So our molecular scissors are going along looking for a GAG. Normally, in the normal gene, they wouldn't find one. And so they just skip right over that because they'd see GAU. Here, because of the mutation, they find a GAG. They say to themselves, oh, a GAG is exactly the sequence that I'm looking for. Snip, snip. They create a splice site there which splices in what we're going to later learn is a stop codon. We're not ready at this point to know about the details of what I mean by a stop codon, but let's just say we put in a stop sign at that point. So in this mutation, because of a splicing problem, because of an editing problem, we end up making an abnormally short message for beta globin polypeptide, which ends up making an abnormally short beta globin polypeptide, which can't perform its normal function. Blood can't carry oxygen normally, and so the patient has a disease. At this point, I have a movie I want to show you of the process of transcription.